Manikam sir, head of the Department of Microbiology, to give a welcome note address, sir. Manikam sir, over to you.
Okay, sir. Most respected principal, honorable chief guest, respected staff members, researchers, scientists, and student friends. Very good morning to all. It gives me immense pleasure to welcome our principal, Dr. C. Ashok sir, for this webinar. His guidance in all our attempts is the fuel to our visions and missions. We welcome you, sir. On behalf of Department of Microbiology and my own behalf, I warmly and respectfully welcome the guest speaker and our alumnus of our department, Dr. Vijayakumar Emory Vaccine Center, USA. I thank him for making his presence despite his busy schedule. We welcome you, sir. Then I welcome to Dr. M. Kanagavel, sir. I also thank him for helping us to arrangement of this webinar. We welcome you, sir. Then I welcome to our Dr. Segar sir, PCG Vaccine Center, and Dr. S. Sivakumar sir, Poison Institute Director, then Dr. Senthil sir, Dr. Venkatesh sir, uh, all welcome you, sir. Then we extend our warm welcome to all the participants, researchers, scientists, professors, students. We welcome you one and all. Thank you.
Thank you, sir. Thank you, Chodi sir, for giving a very lucid welcome note. Now
Thank
we
you,
will
sir.
request Professor Nalini Madam, Assistant Professor, Department of Microbiology, ANJAC, to formally introduce the speaker for the day. Over to you, Madam.
Okay, good morning all, and this is Nalini, Faculty of Microbiology from Ammal College. I feel pleasure to introduce our chief guest. He is Dr. Vijayakumar Velu. He is our guest speaker. At present, he is working as Assistant Professor in Department of Microbiology and Immunology, YerKeys National Primate Research Center, US, from 2014 to till date. And regarding his qualification, he has completed his BSc degree in Zoology in Madras University, PG Microbiology ANJAC. He is an our alumnus. He done his PG during 1998 to 2000, and he has completed his PhD degree in Madras University and postdoc in Emory University during the period of 2005. And then at present, he is a member in different forums like Indian Medical Association, American Society of Microbiology, American Association of Immunology, Theonomics International Private, Bangalore, and he is also a scientist committee of SRM Institute of College and ANJAC. He has received a lot of awards, particularly highlighting we have got Best Thesis Award during his PG, PhD as well as his postdoc. He has received Young Scientist Award, Young Investigator Award. His focused research area is HIV vaccine. He has done a lot of research works in HIV vaccine, and he is also doing some research in antibacterial immunity, particularly respective to Mycobacterium tuberculosis. And he has published nearly 77 research articles, both in reputed journals and in review articles, and he has made a book. And then he has been serving as a special invitee on COVID-19. Recently, during this 2020, he has delivered special lectures from March till now in MGR Medical University, Indian Medical Association of Microbiology.
Thank you, Madam. Thank you for the kind introduction. It is indeed a pleasure to host Dr. Vijayakumar Velu sir from the Emory University YerKeys Primate Research Center. Sir, I think you can now take over. Please share this your desktop and start the presentation, sir. Over to you, sir. And I am muting all the faculty for the time being, except Vijayakumar sir, with your permission.
Hope I'm sharing, right? Hope you can see my slides.
Sir, you are okay, sir? Please start your lecture, sir.
So at the outset, I would like to thank the principal of the ANJAC College for inviting me to give a talk on COVID-19 vaccine development. And I would like to thank Shiva for contacting me initially, and then Dr. Nalini, and then Dr. Pon Manikavel, and everybody for giving me this opportunity. So I'm indeed very much pleasure to give this talk because I'm an alumni of ANJAC. And in fact, for the past 2 days, I was thinking about ANJAC days, how we spent and how we studied the microbiology and all those stuff. And thanks for all the mentors who actually gave us a shape and then for educating people in a very good way. Thank you so much. And same time, I would like to thank Dr. Kanagavel. He's a pillar of this particular program where he really, in his busy schedule, he actually wanted to help us, particularly for this webinar, to host and YouTube as well as arranging many different faculties to participate in this program. So what am I going to today? Today is like, I'm going to talk about the current situation in the vaccine development. I'm an assistant professor in Emory Vaccine Center and faculty in the YerKeys National Primate Center, where we do a lot of monkey works. And this particular center belongs to Emory University, Atlanta. So this is the outline I'm going to talk. First, I'm going to talk about the brief history of SARS-CoV-2, COVID-19, clinical epidemiology and transmission, clinical profile of patients and clinical presentations. And I'm going to touch base little about the therapeutics, where the therapeutics have been developed. People are actually developing these therapeutics against COVID-19. And I'm also going to talk about development of vaccines. And I'm going to talk about a success story of vaccines, COVID-19 vaccine in monkey model. A couple of monkey models have been presented, and I'll show some data on that. At the same time, I'm going to talk about the vaccine that are in clinical evaluation, as well as a preclinical evaluation. So, brief history of SARS. This is the first SARS in 2002, where came from China. This is believed to be a cross-species transmission. We believe that this could be from bat to human, or bat to intermediate host, and probably from there it would have got into the human. So this particular infection, they infected, globally 8,000 cases have been infected, and almost 774 death has been reported, and 32 countries have been infected with this particular virus. And then it stayed for almost from November 2002 to July 2003, almost 8 months it was there. And again, it's believed to be the cross-species transmission. So this is the novel coronavirus, and we have 7 coronavirus, out of which 4 usually causes low pathogenicity. Usually they infect kids mostly, and then the other 3 causes higher pathogenicity. As you can see here, the SARS-CoV, MERS-CoV, and then the current SARS-CoV-2. All these 3 causes high pathogenicity. As you can evident that these 3 actually made a huge hit on the population and a lot of death rate. And the illness is called COVID-19. It's caused by SARS-CoV-2. The reason why they gave this SARS-CoV-2 because this actually more like the SARS-CoV than the MERS-CoV. That's the reason WHO named this SARS-CoV-2. So it is believed from bats. Started late in December 2019 in China, from Wuhan, China, and 25% distinct from SARS. The earlier SARS, about 6,000 nucleotide different from SARS, represents a new clade, very different and indicative of a brand new virus. And again, I see some people are talking about this is a strain. This is not a strain. This is a brand new clade actually. So it's a brand new virus, possibly through cross-species transmission from animal reservoir to a human. Then from China, this particular disease jumped into the world. As you can see, in March 12th, WHO announced this particular disease as pandemic. Almost 45 lakhs people, greater than 45 lakhs people have been infected worldwide. And then almost 3 lakh, greater than 3 lakhs people are died because of this particular virus. Almost 230 countries have been infected with COVID-19, with 84,000 as of now, as of today, 14th, as of yesterday, 14th 2020, 84,000 cases were found in India, and then greater than 2,600 deaths were reported in India. So I just wanted to clarify that, the very first time when I gave a talk, that was March 17th. That time USA had 3,000 cases, but in May now currently they have like close to 1.4 million. So meaning, within 2 months it spreads that fast. So we need to be careful. We have to be careful, particularly in India. Clinical epidemiology and transmission. Again, this is a clinical prognosis and the recovery. Almost 80% of the people usually get recovered by this when they get infected with this virus. Could be because of the immune system. Out of the 40 percentage was without pneumonia, and 40% with moderate illness with pneumonia, and then the 20% that is they really require this oxygen supplement. They are pushed into a category called severe illness and critical illness. And then the critical people who have the underlying condition, I'll talk to you about that underlying condition. Now, those people actually have the septic shock that is called severe acute respiratory distress syndrome. Once it comes, you'll see a massive organ failure. So they die because of massive organ failure. So mostly 80% will be immune. The 20% is the one that is caveat, where when they have an underlying conditions, they're prone to die, they're prone to death. So this is the common symptoms of COVID-19. Predominantly, you see fever in them, and dry cough and fatigue. These are the common symptoms, and some of the other mild symptoms are here. Usually, almost 50% of the case, more than 50% of the case will see these three symptoms mainly. And then it affect people with underlying medical conditions such as hypertension and diabetes and cardiovascular disease or lung disease. So you can see here the critical people, they once they go to the critical, when you see them, those people have these types of underlying conditions. So the patients with these conditions are vulnerable and maybe become a severe and critical easily. So we don't know right now. People from moderate, they go to severe, from severe to critical. Sometime people from severe, they come back to moderate. It's a play between the immune response and the individual's immunity without any underlying conditions. So this is the age distribution and the case fatality rate. This is again, this is a case from study from the China, where you can see that greater than people who have age greater than 60 years mostly were highly vulnerable for the critical conditions, and you can see that the death rate is very higher in that particular age group population. So again, when you talk about children, we don't have much data on children, and data keep coming, but looks like based on the studies, even children are also getting infected, but then they likely to be asymptomatic people. So not much data is available. Probably literature will teach us something. So again, 80 greater than 60 years of age are at high risk, and because they also have the less immune response, and then more of underlying issues. So what we learned so far with COVID-19, this is just to compare between SARS, MERS, and SARS-CoV-2. I would like to concentrate on this particular highlighted part, where the super spreaders are there in this particular SARS-CoV-2. There are many examples for these we have seen. And then the super spreader, they come into the category of the asymptomatic people or a mild spread. So they are the one who's actually spread it very quickly. And then the problem that we are facing right now is because identifying the asymptomatic people. So that's the reason we should actually have a rapid test to screen people serologically to figure out who is coming under the asymptomatic category. And again, the asymptomatic is a big problematic in this pandemic. because if they are super spreader meaning, the people who actually transmit the virus more efficiently. If they exist, if they super spreader exist, meaning 20% of infected individuals, that is super spreader, is responsible for 80% of transmission. So this suggests that a Gaussian should be taken care of asymptomatic people, and this could lead to a rapid spread of the community. So we need definitely rapid testing. It's important to screen people in terms of serology, so that we can go and confirm them and make sure that we quarantine them. Again, natural history, a diagnosis, almost 80% are mild and moderate. As I said earlier, 15% are severe, 5% is critical. 10 to 15% of mild and moderate cases become severe. Again, 15 to 20% severe become critical. So average time of symptom, the exposure to symptom onset is 5 to 6 days. From symptom onset to recovery from mild cases is 2 weeks. And for severe cases, 3 to 6 weeks. From symptom onset to death, it takes like 2 to 8 weeks. Truly asymptomatic infection is unknown without serology, but appears to be rare using molecular testing, less than 1%. An estimated 75% asymptomatic cases at the time of diagnosis soon progress to disease at this scale. Again, there is no much data. This is again the natural history data that I derived from China data, where they don't have a data on the children, because during that time the school was not there. So still data is needed from children to really quantitate the incidence of the virus. This is the virology. The virus shedding is highest early in the course of disease, which peaks at 5 days after the onset. Virus shedding can occur within 24 to 48 hours prior to the symptom. Again, this is very important. This virus shedding can occur prior to the symptom. So the spreaders are mostly asymptomatic. Virus can be isolated from stool, but there is no epidemiological evidence of fecal oral transmission. I think studies has to really come to see whether is there a fecal oral transmission is there. Virus shedding usually continues for 7 to 12 days in mild and moderate cases, and greater than 2 weeks in the severe cases. Patients who recover can be PCR positive after symptoms. And then again, this is again a problem. If we send somebody saying that they recovered, probably they might still be PCR positive. So we need to be really careful. And then you would have to take like close to 28 days to screen them with multiple sampling to make sure that they don't have the PCR positive before they discharge. So COVID and high-risk groups, again, the comorbidity and advanced age increase risk for severe illness and death. Again, cardiovascular is one of the underlying condition, diabetes, chronic respiratory disease, and immunocompromised individuals like people who have HIV. And then again, people who have cancer, they all are coming under immunocompromise. They are at the greatest risk. And then pregnancy, there is no much data on pregnancy, but the thing is, like, it looks like there is no difference between uninfected and COVID infected maternal morbidity. So still data is coming, and I'm actually working on mother to child transmission at Emory. We will know soon what would be the fate of the children, the infants, that they carry the virus or not. Maybe the studies will clearly tell us. Clinical profile of SARS-CoV-2, COVID-19. Again, the cause of death during the SARS is there is the receptor. The virus binds to the ACE2 receptor, and when they bind, actually these particular receptors are high in capillary lung, blood vessels, and lung epithelial cells. They express high levels of ACE2 receptor. When the SARS spike downregulates the ACE2 expression, and which activates the angiotensin receptor. So when the SARS spike binds to the ACE2 receptors, it activates the AT1 receptor, so which results in severe acute respiratory distress syndrome. And when you have that particular syndrome, so that really makes you a big problem, because of this particular receptor expressed in most of the tissues. and different organs. So you will have a massive organ failure when you have this particular syndrome. And then this is the characteristics of patients with COVID-19 infected with pneumonia in China, where this is a JAMA article where they studied 138 hospitalized patients. Here they compared survivors versus non-survivors. You can see the non-survivors in yellow. You can see that the non-survivors do not keep the good lymphocyte count. So there is a lymphocytopenia is happening. They have a reduced lymphocytes compared to the survivors. Again, the D dimer is highly elevated in the non-survivors, and this is an indication of a critical stage of individuals. Again, blood urea nitrogen is higher, very highly elevated during the critical condition. Again, creatinine. So these are kind of a biomarker suggests that immune activation could be a predictor of critical cases. Again, all these markers also tells that there is a massive organ failure. Clinical presentations other than the lung disease, there are a lot of cardiac presentations has been reported without the respiratory symptoms because ACE2 is also expressed highly in the heart. Probably that could be the reason. Then conjunctiva is an alleged route of exposure. It may cause ocular signs. Again, conjunctiva also express high levels of ACE2 receptor. Many abdominal discomfort has been recorded. Viral RNA is detected in stool, and ACE2 is expressed highly in the gut. There are a lot of data coming up that this particular expression is highly in the gut. RNA is detected in saliva. Possibly salival transmission might happen. We don't know. Studies have to prove that as well. And liver enzyme could be a predictor. Again, in the earlier SARS, the 2002 SARS, almost 60% of the patients had a liver impairment. So this could also be a problem. Could be a problem for SARS-CoV-2. What are the countermeasures for the SARS-CoV-2? Again, when you talk about countermeasures, there are therapeutics as well as vaccine. What are the therapeutics available? Again, we all know that the hydroxychloroquine, this is an anti-malarial drug and reasonably strong against MERS and SARS-CoV-2 in vitro culture. And again, one small study in humans against SARS-CoV-2 from France shows some encouraging results, but actually not well-controlled study. It is with a very small population. And it did not work against the SARS-CoV-1, that is the 2002 SARS. Thought to inhibit early stages of virus entry, but the biggest problem for this particular drug is that they have many side effects like blurred vision, nausea, vomiting, cramps, headache, and diarrhea. So again, just yesterday, NIH announced that they're going to conduct a clinical trial with 2,000 patients with hydroxychloroquine with plus azithromycin in COVID-19 patients. Probably, that gives a good data. We have to wait and see whether that is working. Again, we all also know that the recently there is a drug is working that is called remdesivir from Gilead, from a US company, where they showed that it reduces hospitalization from 15 days to 11 days. So if one is not taking the remdesivir, they still stays in the hospital for 15 days. But when they're treated with remdesivir, they're actually discharged within 11 days or so. That definitely will help the economy as well as the benefit for the patients. So some clinical benefits also there, but still they're planning to conduct a massive study with different combination therapy as well. And this is a nucleotide analog, incorporates into the nascent viral RNA chains and result in premature transmission. Termination of the virus. So again, convalescent plasma, individuals recovered from the infection, actually that plasma can be used as a therapy. Again, this is a very old, century-old method. But the problem is we have still unknowns whether the infected individual produce effective antibodies. And then we don't know how the antibody titers maintains in those individuals. And again, the big issue is the scaling up, finding those patients and getting plasma and giving it to people. Scaling up is very, very difficult. This particular paper is published in JAMA in March 27th. Development of vaccines for COVID-19. Again, as I said earlier, this is a new virus, so different clade, brand new virus. So we would have to have a new immunity and a new therapy and a new vaccine for this particular virus. How we develop immunity. Again, in the left-hand side, I'm showing how naturally one can get infected and get immunity. For example, when the spike protein binds to the ACE2 receptor, they internalized, and then the viral RNA translated into proteins, and then the viral assembly will happen, and the viral will get released. Once the viral get released, dendritic cells takes the virus, chop into pieces, and then give it to present it to T cell and the B cell. The B cell make antibodies. If you have a good antibodies, probably you may get protected, meaning you may get the viral clearance. At the same time, if you have a cytotoxic T cell, they also kill the virus. So this is how natural infection will go with a very good immunity. So the adaptive immune system can learn, recognize the virus through vaccines. Since we don't have the vaccine, we would have to educate the immune system to get immune response to this particular virus. How do the vaccine works? Often a weakened form of disease is injected into the body. Some vaccines are not injected but inhaled. Different routes of vaccine can be given. The body thinks the weak virus is threat. It builds a lot of antibodies, particularly the B cell response. Again, this body also may trigger a T cell response. The combination of T cells and B cell will be helpful to attack the virus when we encounter the virus in the second time. So the antiviral B as well as T cell memory is very important to really combat this virus. So we would have to educate, as we all know that we have a lot of vaccines available for different diseases. We are educating the immune system so that the immune system can recognize the virus and then they kill them, and so that we can get protected. So the spike protein of coronavirus is the major target of vaccines right now. As you can see, this is a spike protein. It's a crown-like structure. That's when it's called coronavirus. So this particular protein is a major target for a vaccine. Any given pharmaceutical as well as a company, they're trying to make an antibodies against this, so that this particular antibody can bind to the virus so that it won't enter to the ACE2 receptor in humans. Again, there are a lot of array of vaccines are being developed for the COVID-19. More than 100 vaccines are being developed against SARS-CoV-2 across the world. People use virus as a vaccine, weakened virus as well as inactivated virus they're using as a vaccine. Viral vector like replicating vector or non-replicating vector as a vaccine vector. And they're also trying to do a nucleic acid as a vaccine, like they're using DNA as a vaccine as well as RNA as a vaccine, and then protein-based vaccine like protein subunit as a vaccine, and virus-like particles as a vaccine. So as you can see, the number of vaccine in the development, most of them are protein-based and viral vector-based. So again, when you talk about the immunogenicity, there are pros and cons. Some vaccine work well, but the safety is a problem. Some vaccine is safe, but immunogenicity is a problem. As you can see here, live attenuated is good in inducing immune response, but safety is an issue for that particular vaccine. Like nucleic acid is good in safety, but the problem is that we don't know whether we get a good immune response. So every vaccine has their own merits and demerits. So virus as a vaccines, people use weakened virus as vaccines. So they, in this case, they conventionally weaken the virus for a vaccine being passed through an animal or a human cells until it picks up a mutation. In this case, the mutation might be less virulent, so that this can be used as vaccine. Again, people use inactivated virus as vaccine. In this case, they inactivate them by using formaldehyde or 8 inactivated method to inactivate the virus. And then at least 7 groups in the world are working on these types of vaccines. Viral vector X vaccines, replicating viral vector as vaccines such as measles, weakened measles can be used as a vaccine in this case. And then non-replicating viral vectors such as adenovirus and MVA vectors. These are very good vectors. Many preclinical model has shown that for a different, for example, for HIV vaccine, they show very good immune responses with the adenovirus as well as MVA vectors. At least 20 groups are working on this vaccine. And again, Emory University is making an MVA vaccine for COVID-19. Again, Dr. Rama Amara's group, I did postdoc with him. He's my mentor, postdoc mentor, and he's one of the world's leading HIV vaccinologists. He's working on this vaccine. I'll show you what we are planning to do on this COVID-19 vaccine later. Nucleic acid can be used as a vaccine. People are using DNA as a vaccine. A company called Innovia is working on this. And then RNA can be given as a vaccine. NIH is partnered with a company called Moderna in Boston, where they're trying to deliver RNA as a vaccine. The only caveat there is there is no proven vaccine in the public based on RNA or DNA. So we'd have to see whether this particular vaccine gives a very good immune response. Hopefully, it should give a very good immune response. At least 20 groups are working on this type of vaccines. And then again, most of these vaccines encoded viral spike protein. Protein-based vaccine, again, people can use protein subunit as a vaccine, particularly targeting the spike protein or M protein. At least 28 groups are working on this type of vaccine. Most of these vaccines encode again the spike protein. Virus-like particle, again, this will give a very good immunogenicity. But the problem is this is very difficult to make. Probably people may get a very good vaccine on this. At least 7 groups are working on this particular vaccine. So this is the vaccine that Emory is doing. This is Dr. Rama Amara. So he works on a different strategy, prime boot strategy. Is trying to do a DNA as a vaccine as well as MVA as a vaccine, priming with DNA or MVA, or either doing an MVA based vaccines for this COVID-19. This particular vaccine is currently tested in mice, mouse model, and then we will get the data soon. We'll let you know soon. So COVID vaccine success story in monkey model. This is the data from China, where they use inactivated virus as a vaccine in monkeys. What they did here is, this is a paper in bioRxiv. Again, this came in Science actually. What they did is they inactivated the virus, and then they gave a vaccine of 2 doses, 3 microgram and 6 microgram, in weeks apart for like 3 doses, like 0, 7, and 14. And then 7 days after the last dose, they gave the SARS-CoV-2, and then they followed the animals for the next 9 days or so. They gave 2 doses, 3 microgram as well as 6 microgram. As you can see here, this particular vaccine made a good neutralizing antibody titer. The titer went up after the second dose, and then you can see that before the challenge, they gave the infection year. Before the challenge, they have a good titer, but then after challenge, you can see that the titer is kind of increasing because the immune response is trying to blunt the virus. This is the data for that particular study. You can see the viral titers in throat swab and anal swab. This red indicates a 6 microgram dose. Blue indicates a 3 microgram dose. You can see the 6 microgram dose actually blunt the virus, particularly in the throat swab, and then there is no virus in the anal swab. Again, if you look at the lung, the red one, you see there is no spike of the virus in the lung. And in the placebo, the green one, you can see they're high in virus in the throat, anal swab, as well as the lung, suggesting that the vaccine can protect clearly. This is in the monkey model. There is another study, actually, just yesterday came. I just want to show that as well. This is a chimp adenovirus COVID vaccine. This is from Oxford, I guess. And then what they showed, they prevents COVID-2 pneumonia in monkeys. In this case, what they did is they gave just one shot of this chimp adenovirus vaccine with COVID, and then 28 days after they gave an infection, they followed the animals for 7 days. Here you can see the clinical score. Again, they have this 12-day here, and then the bowel fluid and then nose swabs. Bowel fluid, you can see the red one. Red ones are the one that is COVID-19 vaccine. See that there is no much virus on in the bowel. Either the, I mean, you can see that here in the bowel. But I would like to caution you. But nose swab, still the nose still have the virus. Again, within the lung, within that cavity and the nasal cavity, we see a different variation of the virus. So still you have a virus, but the viral get eventually controlled. So they also show that this prevents a pneumonia in monkeys. So vaccines that are in clinical evaluation at present, this is based on the 11th May. More than 110 vaccines are being developed against SARS-CoV-2 by various research teams around the world, and almost half of them are from China. And then this particular RNA vaccine is from NIH and Moderna. They are making it. They are in the phase 2. This is from adenovirus from China. This is in phase 2 right now. And then they're also using inactivated virus as a vaccine. They are in the phase 1 and 2 currently. And this is a chimp ad. This is in human. This is by Oxford. They're trying to develop this. And again, these guys are trying to collaborate with the Serum Institute of India to make this vaccine in a huge production. So probably it will be helpful to really make a vaccine for a lot of individuals. Again, the RNA and DNA are being used as a vaccine. These are the eight vaccines that are in a clinical trial right now in different phases of vaccine trial. Again, to make the big story short, until the therapy or a vaccine is available, I think we should make sure to adhere to the public health measures, and the need of hour is to case isolation management, and we have to isolate people, and we have to identify people, isolate them, and in fact, I mean, again, we have to quarantine the close contact, and again, we have to follow the strict adherence of suspension of public gatherings, as well as movement restriction like travel. So if we do so, probably we may blunt the curve. Again, India, we have very less infection, but at the same time, we have to be very careful that we shouldn't allow this particular less infection to be a community transmission. So we need to be very strict on public health measures. We would have to follow that. So with that, I would like to acknowledge people here. I would like to acknowledge the whole ANJAC group, entire ANJAC group. Thanks for Dr. Ashok, principal of the college, for inviting me. Dr. Cohen, Body of Microbiology Department, Dr. Nalini for organizing this, Shiva, Mr. Senior, thanks for organizing this, Shiva. Again, special thanks to Dr. Velu. In spite of his busy schedule, actually, he's trying to help us to make this webinar very successful. And I would like to thank Dr. Rama, and most of the slides have been used from him. And even post-document, I would really thank you for all his help. Again, I would like to thank the presentation. Most of the presentation are the literature search. And I got some slides from the Troy presentation. Again, again, I'd like to thank my center for keeping me as an assistant professor. Thank you. So I'm happy to take any questions, and you can email me if they have any questions.
Thank you, sir, for.
There is an audio, some audio problem is there. I can hear.
Sir, please stop sharing, sir, your screen. Thank you, sir, for giving a very, very lucid viewpoint on what is happening on the vaccine side. I hope all of us and the people who are close to around 300 and plus people are watching on the YouTube channel are also very happy to listen and understand what is happening in the vaccine world. So with that, I think we will use your expertise for the benefit of the participants. So may I request Principal
So
Sir?
there is a sound problem actually. I'm hearing. Uh,
Let me try, sir. Is that all right now, sir?
yeah, now it's all right.
No, I think I'm unmuting. So I will request all the faculty who expect whoever is speaking to unmute themselves so that sound issues cannot come. On this behalf, I would like to request the Principal Sir, Professor Ashok Sir, to give your viewpoints and please share your thoughts if any, sir. Ashok Sir, over to you.
Sir, Ashok sir is not here. Let us start from
HOD sir, Ponmanikam sir.
HOD sir. HOD sir can share.
We got more information, your presentation, sir. We got more information. Sir, some of the countries are trying herd immunity. What way that herd immunity is beneficial to their people, sir?
So the thing is, again, the Kalanjar TV பேசின மாதிரி herd immunityன்றது வந்து we have to infect or we have to vaccinate at least 70% of people population. அப்பதான் வந்து we should talk about herd immunity. With this, again, we have 120, 130, 120 crore people in India. So you have to vaccinate 84 crore people. Then only you can talk about herd immunity. So it's premature to talk about herd immunity. I don't think that's happening. That's going to happen right away. So it will take some. We'd have to have a vaccine for that. Then only we can expect a herd immunity.
Okay, sir.
Thank you, sir. Nalini madam, can you give your comments and you can ask your question, madam. Over to you now.
Good morning, Vijayakumar sir. Its presentation is so nice. We got a lot of information. I do have a doubt that I heard in a news. I studied in newspaper that there are some antiviral like remdesivir and favipiravir. Is it effective to the patient, or is it under trial? I heard in a newspaper that remdesivir is going for trial for some patients. Why it is not efficient? Do you have any idea regarding this?
No, we don't know that.
So can you hear me?
Yeah, I can hear you.
Ah.
We don't know that.
Remdesivir antiviral. So
Yeah. So as I said in my presentation, I think studies are conducted in remdesivir is like very poor population, and probably we need to have a more number of population this drug. So many studies has to come. Then only we can really say that whether it is working or not. But looks like, as I said, it's actually reducing the hospitalization at this point. Remdesivir. There is no any study for the other drug right now. Probably, soon we may hear it. We have to wait. This is in the early stages of the studies. We'd have to do a large number of groups so that we know what exactly happening. So there are studies coming. It's not working. Some studies saying it's working. I think we would have to conclusively see many studies or many different populations to see whether this is working really well or not. But at this point, they are recommending remdesivir in US, and US is trying to make a combination therapy kind of stuff for remdesivir, and probably we'll know once that particular trial is over. So we'd have to wait and see for both the drugs.
side. That is the genome. There is a report that the genomic sequences has some mutated. There are different strains of virus. In India, there is a different strain, and China, where there is a different strain. So, does the treatment and vaccine suits for all these different strains?
Yeah, definitely. I think it is not like mutating like influenza. So it's not mutating like influenza. I think the mutation rate not that really vast compared to influenza. I think the vaccine, if the vaccine has a very good neutralizing antibody, any vaccine probably that might help in different strains for this particular virus. The first thing is to get a good vaccine and a good immune response.
Thank you, sir. Thank you, Nalini madam. Now I request the department faculty, Dr. Shivakumar sir, to ask his question, please. Shivakumar sir? Okay, I think he has some challenges in his audio. Can I request Maheshwari madam to ask her question, please? Please unmute yourself and speak, madam.
Sir, how to protect the low immunity person? Any special treatment available?
No, at this point, there is no treatment. We'd have to India வந்து இப்போ வந்து they were prescribing some Ayurvedic medicine or I don't know Ayush kind of stuff. We have to try certain things, but again நான் வந்து என்ன சொல்வேன்னா we would have to be very careful. Just like that எதுவுமே எடுத்துக்கூடாது. We don't know what's happening inside our body. That may aggravate as well. So we would have to be careful. I cannot say anything right now because there is no any proven study. As a scientist, I think we should be very careful recommending anything to people. Just like that, without a study, clinical trial, we cannot take just anything like that. You know, that is not at all appropriate.
Okay, sir. Thank you.
Shivakumar sir?
Yes, sir.
Please
Yes, sir. Okay.
unmute
Okay,
yourself
okay.
and ask your question, sir, please.
Okay, sir. Okay. Hello, Vijayakumar. Nice presentation.
Thanks, Shiva.
Okay, two more questions. Simple question. Why our Indian people is highly resistant than foreign people? Because of death rate is very, very small when compared to foreign countries. After 2 months lockdown. Okay, what is the reason behind that? Is there any immunity?
No, I think people are talking. The genetics could be different. அதை பத்தி பேசிட்டு இருக்காங்க. Climate is different. அதை பத்தி பேசிட்டு இருக்காங்க. But nothing has been proved yet. Again, people are talking about BCG vaccine. United people அதிகமா இருக்காங்க இந்தியால. So, but BCG trial is going on with COVID-19. But until or unless any data comes, peer-reviewed data comes, we cannot say at this point. But thank that we didn't have much infection compared to other part of the world. Illana, we can't afford this. So again, prevention is better than cure. We'd have to prevent. So we have to stay home to not to get the infection. So the strict, if we follow that, probably we may really dampen the curve, and it will be very. If it's spread, it's going to be as I told when I was talking very first time about COVID-19. That is, I think March 27th or 17th. That time US only had 3,500 cases. But today, what? May 15th, US has like, what? 1.5 million, 1.4. So think about it. So it'll spread that fast.
Okay.
Yeah.
Whether it is
next question, sir.
So whether it is a synthetic or not, some of the people told that it is a synthetic.
No. There is no clear study saying that this is synthetic virus. No.
Okay.
It's
Right, sir. Thank you.
Thank you, Shivakumar, sir. Now I like to call Vijayashanti madam to unmute herself and request her to ask the question, please.
Okay, sir. Good morning, sir. How is the coronavirus spread by infected people who have no symptoms, sir?
Right. So that's the peculiarity of this particular virus. So since the nasal, that particular route is the one most efficiently transmitting, that's what we don't know. This could be that particular virus, that particular droplets might have a lot of virus in it. You can see whenever they detect the nasopharyngeal swab or something, you see a lot of the CT values. You can, based on the CT value, you can tell that they have a lot of virus. So probably the infection rate is high because that particular those droplets might have a lot of virus. We don't know it clearly, but then look at age-wise analysis. As I said, the fatality rate as well as the adults and then the adolescents or the kids. Adults and adolescents might have a very good immune response. They might keep the virus into the, like, they can control the virus, but at the same time, initially during the infection, they could easily spread the virus to the elders. So the elders get very, like, when they have greater than 40, 50 years old, they can get the virus. They might not have a good immune response. So these all combination factors. The viral load is droplet viral load is a factor. Health is a factor. So all these will affect. Again, this is a new virus. We have to have a new immunity.
Vijayashanti madam, your second
Okay,
question, please.
Sir, no, sir. Thank you, sir.
Thank you, madam. Thank you. Now I request Dr. Mahendran sir from ANJAC Microbiology to ask his question, sir. Mahendran, over to you, please.
Sorry, sir, nice speak, sir. Very useful one, sir. Sir, my question is, COVID-19 single standard RNA virus or double standard RNA virus, sir?
RNA virus. Is that the question?
Sir, he would like to know how many strands of RNA are there, sir?
DNA.
I think this is single stranded RNA.
Mahendran sir, any other questions, sir?
Okay, thank you, sir.
Thank you, Mahendran sir.
No, sir.
Now, thank you, sir. Now we request Munusamy sir to unmute himself and ask the next question, please. Munusamy sir.
Yes, sir, it is a nice presentation. He narrated the current scenario of COVID-19 vaccine development. In an article, they reported that angiotensin converting enzyme 2 is mostly affected by the COVID-19. Is there any drugs available for this boosting of this angiotensin converting enzyme, sir?
Any drug available? There are a lot of drugs available. But we don't know as of now, until or unless there are a lot of drugs available. There are naturopathy also. There are a lot of food products, been shown to see increase this angiotensin system. But then we would have to try. At this time, this is just like 5 months old virus. We have to try. Without that, I cannot comment on it. But there are a lot of drugs to inhibit the ACE2 receptor as well. But until or unless we do the proper clinical trial, we cannot say anything.
Okay, sir. The next question is that the youngsters, that is the below age of 3, they won't affected by COVID-19. Is there any reason or any reports are not available regarding this one?
So the thing is, I think there are a lot of reports needs to come. We don't have a clear report on kids right now, but there are beliefs that other coronaviruses that I showed you in the less pathogenic viruses might have
4
infected them. That might have occupied the ACE2 receptor, because that might not allowing this one. This has to be showed clearly. We don't have the data right now because people are talking this could happen, but until or unless we know a clear study showing that particular pattern, we cannot say anything. But again, we all know that the other coronavirus, we all are exposed to other coronaviruses, right? Like it'll also have a flu-like symptoms. So we gone through that, but then if the kids has more of that, probably that might not allow the real COVID too.
COVID-19.
Myth and the belief that people have, but probably until or unless we have a study, clear pre-reviewed studies available, we cannot say anything right now. Again,
Okay, sir. Thank you. Thank you.
that could be a natural thing that kids can really protect. We don't know until or unless it comes. We cannot say anything.
Okay, sir. Thank you, sir. Thank you, sir.
Manuswamy sir, thank you so much for your question. Now we move on to Dr. Sivakasi Narayani madam. Please unmute yourself and ask your question, madam.
So thank you, sir. Thank you for your nice
Yes.
presentation. Sir, can you hear me, sir?
Yeah, I can hear you.
Okay, sir. Sir, thank you, sir. Actually, my query is that any drug which is induce the cytokinins, cytotoxic T cells, NK cells, B lymphocytes can be used as a treatment, right? Any
Right.
type of compound.
There are a lot of clinical trials going on in this
Oh,
time. Because of this pandemic, people are trying different possible ways.
Okay.
People are also trying checkpoint blockades.
Okay.
Checkpoint blockade meaning like the PD-1 blockade, certain JAK-STAT inhibitor. These are all in the clinical trial. People are trying to really trying to use drugs that actually reduce inflammation to see
Okay.
whether
Okay.
it's effective or not at this point. I think
Okay.
that also help actually.
Sorry, sir, I can't hear you. Hello?
Yeah,
Hello?
that will also. I can hear you properly. So that will also help. Any drug that actually put down the inflammation, probably that might help.
Oh,
But
okay, sir. Okay.
we'd have to
Work on it.
right until or
Work on
unless
it.
do and study, we cannot tell just
Give
like
that.
that this will work or
Oh,
not. Again, the
okay, sir.
checkpoint blockade. Our lab is one of the pioneered in checkpoint blockade on HIV.
Okay.
We feel, probably if we break that exhaustion, probably that might help. The question
Okay.
that you asked might be the NK cell, T cell, B cell can actually get the strong immune response, and then probably they can help actually. So we were thinking on that direction. We are trying to get a grant on that.
Okay, sir. So next question, sir. Hello, sir. Hello.
Madam,
Hello, sir.
please continue to ask the question, madam.
Okay, sir. Sir, actually, is that any possible to arise reinfection for the same person?
No, I think right now there is no data on the reinfection.
Okay.
Until or unless if you do a false positive result and then false negative result, then if you come with the fourth day or fifth day with the correct positive result, I think that's what happening right now. There is no clear data that there is a reinfection for the matter of note. In monkeys, they re-challenged it. Nothing happened. The monkeys
Okay.
they did a re-challenge and nothing happened. So they
Okay,
did not
sir.
inspection.
Okay. So, but can we prefer that vaccine from the recovered human beings?
Right, that's the thing. Like plasma can be
That's the
medicine, or else
yes,
people
sir.
people can sort B cells and then sort
Okay.
plasma blast and make a antibody out of it. People are trying to do it, and Emory is going to do it that as well.
It's all are available in market right now, sir.
Which one?
That human-based vaccines.
Nothing is in market right now.
Okay, sir. Okay, sir. Okay, sir. And another one doubt is, sir. So, how can we identify this virus as a weakened one? Weakened virus can be used as a vaccines, right?
Right. That's what in the Oxford trial where they were using
Okay.
the Chinese trial, they used weakened virus as a vaccine.
Okay.
It looks like they make a good T cell and B cell response. Probably that will work. Again, we would have to have a proper human clinical trial for that to decide. So
Sir,
these
actually,
are.
hello, sir. Hello.
Madam,
Yeah.
please ask your question, madam.
Okay, sir. Actually, sir, what is the Vaccinia ankara? Modified Vaccinia ankara.
Yeah, this is a vaccine. They passaged it so that they took away all the pathogenic genes of the vaccine. So that's what it called the modified vaccine ankara.
Okay, it was combo with DNA prime and it can be used as a booster.
Right. So right now we are trying to have actually my boss, as I mentioned, Dr. Ammal is working on it, and we have a vaccine for HIV. We are trying to do this in monkey model. This looks good in monkey model. Probably
Okay,
be the same for human as well.
Yes, sir. Okay. So thank you, sir. Thank you so much.
Thank you, Shankar Narayan, Sivakasi Narayani, madam. May I request Dr. John Wilson, sir, to ask his question, please?
Yes, sir. So it's me, Dr. John Wilson. Good morning, sir. So my question is a plasma therapy done by use?
What is it?
Plasma therapy done in use?
Yeah, yeah, yeah. Yeah. They're actually particularly in the Dr. Kanagwe will know this, because of that. அவங்க பேர் நான் மறந்துட்டேன். அவங்க
Professor Muttalagu from Mass General, UMass.
UMass. I think she is actually doing it. That group is doing it actually. So they're actually contacting the recovered people and getting plasma right away, and then trying to infuse those plasma to the critical patient so that they can get rid of the virus. So it's actively going on right now, except the fact that the people who got infected have to come forward to give the plasma.
Again, so what's the success rate? Success rate of plasma therapy?
I don't know that, right? I don't know the data yet. So I can't see the success rate. But again, we have to be very cautious because we don't know whether the infected people has the right amount of antibodies to really dampen the virus in the other host. So we need to be careful on it.
So what the question is?
After she was telling that she has not looked at the titers, right? Dr. Kanagwel, sir?
Sir, to briefly chip into this question asked by the faculty. Sir, it is still considered as a very limited trial. And
Right.
the trials are approved for already people who are doing plasma-based therapy for leukemias and other things, where the non-responders to the conventional therapies are only given plasma therapy at this point of time.
Right.
Still, as you rightly pointed out, the desired concentration or the desired immunogenic substance from the donor has been not very clearly quantified, which segment of the plasma is truly helpful. Because the challenge which very recently, yesterday we were discussing was having a new plasma that is apart from the respective COVID immune helper. The other things also pose a challenge to your already immune challenged person who's getting the brunt of COVID-2. So they are telling we have to regionalize the donor plasma to a very specific zone, or probably at that particular bandwidth of group of plasma alone has to be segregated. So at this point, they are working on diversification of the zone of selection. That is what is the very recent information, sir. But I am sure it is not still going to be a very uniformly approved therapy at this point of time, sir. John Wilson, sir, I think you can ask your question. I think you have one more question to go.
Yes. One more question is there any positive correlation between the temperature and the COVID positive, sir?
I don't think so. Again, there is no clear studies available. See, the thing is like the droplet might be dehydrated once you have a high temperature, but then there is, again, studies are not there to really clearly point out. Studies are there to show that they spreads and they stays in the air for a while, as well as they stays in the different surfaces, like in papers or in the plastic surfaces and metal surfaces and all. But the temperature study has not been shown clearly. So again, we need a lot of studies to come to really get to know about this virus.
Thanks, sir. Thank you, sir.
Thank you, sir. Now we have two important guests. So because the host institute takes the priority, we finish with the faculty of the host institute. We have two senior person who has been with us from the beginning. We have Professor Raghu from the BCG Research Lab from Chennai and Professor Shivakumar from the Pasteur Institute, Kunur, we have on board. May I request Professor Raghu, sir, from BCG Lab, Chennai, to give his comments and ask his questions, sir. Over to you, sir, please. Sir, please unmute yourself, sir. I think we have some challenge, sir. I will go to Director Pasteur Institute and come back to you, sir. Please allow me to do that. Shivakumar, sir, from Pasteur Institute, can you please unmute yourselves and ask your, first give your comment and then you can ask your question, sir.
Thank you all for this wonderful opportunity to participate in this webinar.
Sir, maybe you need to come little closer, sir. Your mic, please.
First of all, I would like to thank your college to allow us to participate in this webinar. Thank you very much. And the presentation is very good. I'm having few questions to Dr. Vijay.
Yeah.
Hello, Dr. Vijay, how are you?
Good, sir. How are you?
We are good. One small thing, actually. My colleague, Dr. Shekhar, he is leading our R&D. He has a small question on this.
I can't hear it properly. So can you be please louder, sir?
Yeah. My colleague, Dr. Shekhar, he is having small questions. So I'll hand over this to him.
Sure.
Hello, sir. Good morning, sir. How are you?
Good. How are you, sir?
Yeah, I'm fine, sir. Yeah, this time clarify regarding that the MED vector, modified vaccine Ankara.
Yeah.
And it's like the S protein of the virus is giving the more neutralizing antibodies compared to others. So it is preferred to clone the S protein alone or the E or matrix protein along with the
I don't know what plan they're right now with the MVA.
Yeah.
I'm not a part of that study.
Okay.
I know about the study, but looks like the first target of vaccine is that they are doing with the S protein right now.
Okay, S protein alone. Yeah.
But again, I may hear more after a while. But then looks like the S protein is the one target right now.
Okay.
Both for the DNA as well as the MVA.
Okay, sir. Yeah, thank you, sir.
Dr. Vijay?
Yes.
Can you hear me?
Enable your video, sir.
I can hear you, sir.
Yes, sir.
Can you hear me now?
Very well. Please go ahead.
In case if you are developing some protein, we want to develop some antibodies experimentally. Is it possible to test at your lab?
Not in my lab, probably in my boss's lab. I have to check with him. Yes. We can.
I'll mail you officially. I will send an official mail.
Sure. Since you are from Passion Institute, probably I can give him give him your address and then exchange those emails. Probably that will help you.
Thank you, doctor. Take care. Thank you.
Thank you, Shivkumar, sir.
Thank you.
Now I think we have Professor Raghu from BCG lab. Sir, are you with us, sir? Professor Raghu, I think he's... sir, are you with us? Okay. I think now in the meantime, there have been few questions which have been asked in the participant group. I'm going to run through the question. So, sir, they want to know how long is the possible mathematical predicted model for this pandemic to get over?
That is critical question right now. I think a couple of days ago, there is some belief that probably this will stay for a longer time. Right now, again, until or unless any intervention like therapy or a vaccine comes, this will stay for a while. That's the prediction right now. I don't know about the mathematical model, but it will be for a while until or unless you get a vaccine or a therapy.
Thank you.
Because people get infected. See, even though with the strict lockdown in India, we never had a lower case, right? We always went up with the cases, and even that is true for US as well. So until or unless any intervention comes, we cannot predict how long this will stay. So yeah, so that's the answer.
There is one question from one of our postdoc, Dr. Meera Amog. She is asking you how human RNA polymerase can cause mutation in the coronavirus. Is that possible, or what is the mechanism behind it?
So I don't know about it, but I actually don't know the answer about it. I'm not a molecular virologist to answer that.
I understand, sir. And then there is a question. Is there to declare a patient cured patient, is there anything like there was a talk called immune passport? So they go on to say, okay, this chap has gained immunity. He's okay to go back and he's not no more harmful to the society as a spreader. Are we doing any work on that? And what is the current update on that, sir?
I think people are talking, different countries are talking about different, I mean, like these regulations and passport and all. But I definitely would like to recommend to check people before they discharge. For example, they would have to be there for at least 28 days. Even in the monkey model, there are certain reports that you can see the virus on 20 plus days. So we would have to check thoroughly with the different sampling procedures, like different swabs, and we have to be thorough that they are PCR negative. Then only we can make a conclusion. But then again, then they have a good immune response, they clear it, but then they won't. We would have to check before discharging them that they don't have the virus. That's through the PCR method with different. Without that, we cannot have a conclusion to discharge them. We'd have to do it thoroughly, not only 14 days. I would recommend at least for 28 days.
Thank you, sir. The next question is from one of PG, Darjili Dharmaraj. She would like to know, can antisense RNA technology be used against COVID-19? Is
Probably.
that used anywhere?
People are trying different aspects. I don't know anybody working on the antisense RNA, but probably studies might come. But as of, I don't know about that particular technology being used in this vaccine or not. I don't know that yet.
Thank you, sir. There is one student from by name Iniya Tamil Tender. She would like to know, what about the mutated strains of COVID-19? Does it mean the to be produced vaccines, which is mainly oriented towards common strain, will continue to be effective for future mutated mutations?
So see, the thing is, mutation go different in both the side. Mild can go to wild can go to mild. So we right again, the lot of sequence studies has to come to really confirm this particular pattern, whether the mutation that or people say that India has a less virulent and all the stuff. We don't know that yet. Based on a sequencing data, we cannot say that it is a less virulent or more virulent. We cannot say that. Studies has to come, comparing different regions, different pockets of the world. Then only we can actually say something evidently.
Thank you, sir. And then one faculty by name Professor Ajita Ravichandran. She would like to know, what are the primary challenges or restrictions which you face currently or you expect to face during the development process of vaccine for COVID?
So there are a lot of challenges, right? Like we always, again, for 40, almost close to 45 days, we are locked down. We are at home. Only with the permission we go to lab and work. At the same time, you are not exposed to the world. Mentally, we have a problem sitting at home and all. There are a lot of challenges. Again, everybody has different types of challenges. At this point, I can say the biggest challenge is to get a vaccine. We would have to get it very quickly, or the treatment, so that it can help people at this point. And then I would like to try different things to really to have an impact on this particular virus. And again, world is doing that. They are trying particularly US is funding in different levels to really get to know about what are the other strategies that can actually work in this COVID-19. So the challenge is really to make a good vaccine or good therapy. Then again, we all are in lockdown. We'd have to be mentally should be stable to combat this pandemic.
Really a pragmatic answer and approach towards this. The next question is, I'm not sure she is a postdoc or a faculty, Madam Kavita Dandapani. She is asking you, can you please tell us about the post-infective immune status? Like people who have cured, okay, are we assured they are okay? Like, or do they still expect to is there a possibility of a recurrence in them also? One cure, is it fine to tell they are safe? That's a preliminary question.
So at this point, I can say that there is no recurrence of infection is happening. but at the same time, there is a paper from Emory in bioarchive where they looked at the system of the individuals who are exposed to the virus. They have a very good correlation between the binding antibody as well as a neutralization antibody, suggesting that at least greater than 90% of them are correlating, suggesting that the exposed individuals are actually generating a good neutralizing antibody. So I think that is a good evidence, and probably they have a very good, exposed people have a good immunity, particularly the B cell response.
Thank you, sir. Then there is a question from Gangadhara Prabhu. So what he goes on to ask is, this virus has started from a sort of a winter country or a colder climate, but it looks like it is moving to temperate and warm countries and cold countries in a different perspective. Is there a rationale behind the spread pattern or the increased incidence in certain countries towards the specific zone? Do you have any thoughts about it, sir?
Again, the temperature question is high. All are like kind of an hypothetical right now, but there is no any clear study on that. And again, when you look at the pattern, it's again compared to December to now, still we have a different pattern of temperature in the country, right? So, but still we have more infection. So we cannot account for a temperature, and I think unless there is a clear data, we cannot say that the temperature is influencing the virus. That is, we cannot say that until or unless we know for sure. So
thank you, sir. Thank you. And then next question is from Dr. Nitish Kannan. Are there any other proteins in the virus to develop as a protein-based vaccine, or only the receptor binding protein is at present targeted for making the protein-based vaccines?
People are trying M protein as well. So there are different. They're working on both T cell and B cell based vaccines. At this point, I think most of the people are working on the spike protein, and then some people are also working. Companies working also for the M protein.
Dr. Shekhar, sir, from BCG lab, are you with us, sir? Shekhar,
can you hear me?
Yes, sir, yeah. Very well, sir. I think your audio is now good. Dr. Vijay, this is Professor Shekhar. He is from the BCG Research Center at the from Chennai. Sir, your comments and your questions, please, sir. Dr. Shekhar, sir.
Yeah. Dr. Vijayakumar, it's a very nice presentation.
Thank you.
And as you are told, can you hear me, Dr. Vijay?
I can hear you properly.
Yeah. As you said, there is an ecological report that BCG vaccination offers some protection, but still there are some experimental work going on in countries like Australia and Netherlands, as you pointed out. But there are, as I know, there are some animals which can be infected. I mean, it can be infected with the COVID. The question I ask is that, as a part of BCG vaccine production, we immunize guinea pigs and challenge with the virulent mycobacteria. I wonder whether that animal model, wherein we immunize the guinea pig with the BCG, what we conventionally produce to attain immunity, whether such model can be used for challenge with the COVID.
Yeah, so I think at this point, the major studies are going on monkeys, different species of monkeys. They're exposing the virus. So one of the good model right now is like marmoset as well as the rhesus monkeys. Again, rhesus monkeys still people are working on it. If that particular virus, this particular virus, infect the guinea pig the right way, probably we can use that as a model. At this point, I don't think any studies shown that SARS-CoV-2 have an established infection in guinea pig. If that is the case, probably we can use that.
I came across one reference in 2005, wherein they used SARS-CoV, possibly SARS-CoV-1, in which they have showed guinea pig can be experimentally been infected. Whether any information is available on SARS-CoV-2, that's what my question is.
I don't think so. I'm not aware of something. I can check right away.
Okay, doctor. Then my next question is that, as you rightly pointed out, asymptomatics are super spreaders. But when we see the strategy of the RT-PCR testing, to find out the COVID positivity. If you take even Tamil Nadu, we do about 12 to 30,000 tests per day. But if you see the positivity, it's only maximum about 4%. Among the 4%, about 90% of them are asymptomatic. And in a country where the population is about 130 crores plus, whether we can do the RT-PCR test for all the population to find out the asymptomatic, which is not feasible operationally, as we all know. And you alternately suggested serological studies can also be done. If that is so, what could be the ideal marker to pinpoint that these people, asymptomatics, can also transmit the infection?
For answering your first part of the question, so the first part of the question is about the infection rate, right?
Yeah.
The first part, right, sir?
Yes, because most of them are asymptomatic, and most of them turn to be negative also, because most of the tests are done among the contacts as in index cases. If that is so, whether the strategy of same RT-PCR can be continued, or as you alternately said, serological tests.
So I think, sir, that there's an issue of economy as well here in this question. So you cannot do RT-PCR for everybody right now. I think they're charging 4,500 rupees or something.
Exactly.
But what we should do right now at this stage, at this point in India, with a massive population, is that we should have an effective rapid test. First, we should have a serological test. You cannot go and you cannot do a contact tracing and do RT-PCR for all of them. Okay.
Exactly.
Rather, what you should do is, if you have a contact people, you should go and screen them for IgG and IgM to know in which level they are in, which status they are in, whether they are carrier or they're a cleared infection or they're asymptomatic cleared infection. So based on the serology, maybe we should move forward with the RT-PCR. I think that is most economical as well. At this point, if you try to do RT-PCR for everybody, it's going to be a big economy devastating thing will happen. I think we have to screen serology first and then go for RT-PCR. Again, RT-PCR is must. I'm not telling we shouldn't do RT-PCR, but the same thing, we have to be selective in doing RT-PCR, not for the contact tracing and all. First, we should screen them with a rapid test or a proper ELISA test.
Okay, sir. Thank you. Thank you very much. Thank you, coordinator. Thank you so much.
Thank you, sir. So I can't hear you, sir. I can't hear you. No,
am I okay?
Yeah, you're okay now.
There is a question from one Dr. Rajalakshmi Chandrasekhar. There are third generation sequencing techniques like nanopore, and they are more specific. Why the world is still sticking towards RT-PCR? Does it have a specific reason to stick on to RT-PCR alone as a tool?
So different diagnostic procedures are evolving, and people are trying to get the fund and try to make it very simpler as well. And I think they are all in the trial run right now. That's a good question, but then somebody has to implement that with a very cost-effective way than the RT-PCR. Then only we can have a solution for this. At the same time, again, even in doing an RT-PCR, not all the labs are equipped with RT-PCR. So government is funding them, giving the machine to them. Probably we would have to do that way for any newer diagnostic inventions. So there are a lot of strategies. People are trying to do CRISPR-Cas9 and all those stuff, and all these things are being implemented. But at the same time, we would have to stick on to something that is really economical as well as cheap, particularly a country like India.
Thank you, sir. Then there is one question from Dr. Akila Beni. Sir, I've heard that the L strain of this virus is more virulent. So what makes this strain more virulent than the other strains?
We don't know that yet. So China had it more, Italy had it more. We don't know what makes them that much virulent. So again, this is a strategy. It's a battle between the immune response versus the virus. The virus that actually infects you will be different than me, but then it mutates internally. So that picks up some. So again, immunity between you and me is very different. So that it's the game between the immune response versus the virus.
Fair enough, sir. Then there is one person, I don't know, I think he's speaking from his department mail, botany applications. Sir, is there any update on phytotherapy? Like the previous therapies which have proven to have some antiviral effects. Are you aware of any published research which are right now pointing towards some beneficial effect of phytochemicals which are coming up?
Phytochemical meaning plant-based, is it?
Correct.
So I think we have done one study. There is a compound called the Karanjiragam solider. So we isolated one particular compound in it. We did some docking results on it. Looks like it's binding to that domain. Like that a lot of, I think if you look at the MGR University, they are also trying to do something like that. Probably all thing will work. And in fact, Philanthus amaris, my boss, my PhD mentor, Dr. S.P. Tyagarajan, he found out the Philanthus amaris for the phyllanthine for the hepatitis.
Yes.
Looks like that also works in this case. But at the same time, until or unless we test this properly, we cannot say anything that it is working. Again, நம்ம party காலத்து வைத்தியம் மாதிரி we would have to do something to make ourselves comfortable during that infection. But my recommendation is எதுவுமே தெரியாம நம்ம எதுவுமே உள்ள போட்டுக்கூடாது. So that may aggravate things, So அதனால we have to be very careful while recommending these types of procedure to people. Until or unless the studies are clearly shown, we cannot say that.
Point is taken. At this point, we do not have replicable research or is a replicable publication which is being
right. I think at the same time, I really wanted, and I think the ICMR has to really boost up these things to make a lot of trials like this to prove that the certain therapy are working very well. Until unless you fund them and make sure you do the active research, you are not going to find anything. So, or else we have to depend on somebody else.
In fact, to share some information, there are a couple of universities being funded by CDC who are right now looking into it. One thing is the specific plant, the combination therapy like Nilavembu Kudineer, which is being promoted by the government. It has earlier proven to improve the overall immunity level. But at this point, they have extrapolated this into broke down like especially the molecule which have been the more active stuff of the Nilavembu Kudineer or any other therapy, Kabasura Kudineer, all those things have been researched, and CDC has funded, and preliminary recruitment is on. In Karnataka, one university, and Uttar Pradesh, there is one university has started up. But right now, the ethical committee has cleared and they have just got into the study. Maybe, as you say, we have to have clear data about it. So even the US government and Indian government are right now funding. And right now, the ICMR has relaxed the funding approval process also in this regard. So people who are interested in doing it should directly contact the ICMR for this. And we move on to the next question.
I see, the thing is, the lot of plant-derived products definitely be immunomodulators for sure. But at the same time, we would have to know which type of immune response that particular product is triggering. For instance, if you think about the SARS-CoV, the first COVID happened in 2002, if that particular virus elicits a Th2 response, then you'll have an hypersensitivity reaction. So we would have to be very careful what type of immune responses this particular drugs, the phytochemical drugs are doing. We would have to study that until or unless, you have to be very cautious doing it. Again, it works well. We are going in the right direction. I know that the Kabashura Kudineer, I was talking to my mentor, Dr. S.P. Tyagarajan, he was telling what are all the compounds there. It looks like 15 different types of herbs are there, but still you have to really know what that is doing. So right now it's well and good, but then we have to be cautious.
Point is taken, sir. Then the next question from the one of the participant, I'm not sure. She goes on to ask, sir, the hydroxychloroquine has been tried at different levels, either as a prophylactic phase. Dr. Akshaya Vijay. So she goes on to say, how effective is hydroxychloroquine from laboratory point of view?
No, I think, yeah. So in terms of the in vitro, probably it is working, but at the same time, it is not working in patients. As of now, there is no clear evidence that this particular drug is working very well against COVID-19. So that's when now the NIH is trying to do a larger study for 2,000 participants. Probably that will tell us, in a combination of azithromycin, probably that will tell us what would be the fate of that particular results. Right now, I don't think it is working.
Dr. Srinivasan Sampath, I don't know from which institute, he is from the material science department. Sir, what is the actual size of COVID-19 virus? Because it is given as a range. Does it mean it has?
It's like if you have a droplet without moisture in it, it is like 4 micrometer. I think that's what micron. That's what I vaguely remember that. I may be wrong, but it's 4 micron without the droplet. I mean, with the moisture, without the moisture or something.
Madam Nalini, madam, I think you have few questions coming up. I think I have another 4 or 5 left with. You can ask your questions and I will wrap up, madam. Nalini, madam. Okay, I think she's getting ready. At this point of time, there is a question from one of the faculty. Sir, rapid test is being advocated at various levels. Korea had a rapid test. Other countries also have a rapid test, especially in the contingency plans which are being raised up to start the air travel, to start international travel as a pre-boarding screening. Like a spot test is being planned by various countries. In fact, the Emirates Airlines have already earmarked places like going through the security itself will be a different experience in going to be the post-COVID travel business. So, do you think if the way the nucleoside research is going, are we directly making a single nucleotide rapid test, which is in the near future, or is it a valid thing to facilitate other human movements as such based on it?
The thing, I think, the rapid test in FD Pakrana, I think it's like a pregnancy test. So, if you wanted to go look for a pregnancy test, it has to be like 99.9% accurate, right? But in this case, it is not that accurate. If they have like 80%, you are missing 20% of people with your rapid test. So that is a huge mark. So we need to have a very good and a sensitivity test for rapid. So then only we can actually implement these type of stuff. At this point, we can't do anything. We keep doing the tests that we have right now, and at the same time, we have to increase funding to get a good rapid test availability. So the sensitivity issue is the biggest concern here, but we have to make sure the sensitivity is like close to the greater than 95% or so. Then only we can really be assured of any strategies. It's like
possibly a screening test will you validate that statement at least, sir?
Right.
Okay. So there is a next question from Dr. Nuruddin Jabbar. Sir, very limited success from Israel has showed the pluripotent stem cell therapy are helpful. Right. What is your take on it? Are you working or do you know any publication based on that?
No, we are again. We are doing something in the cord blood and then trying to get something. I'm not doing actual stem cell research, but at the same time, looks like that is a promising strategy. The problem there is going to be the cost. Keeping a cell line or cells for a while, it's going to be very cost. mean, the cost is a matter. It's going to be an expensive therapy. Again, you cannot implement to all the patients through that. Growing a stem cell itself a big thing. So that is an effective one, but at the same time, you cannot implement to all the patients. So we would have to, the vaccine is the best solution for this. We would have to have a good vaccine. Vaccine comes, then we'll have a very good chance. Again, therapy-wise, as I said earlier, the vaccine is the one to prevent infection. Therapy is the one to give it to people who already got infected. We have very less people around the world got infected, but the uninfected people are a large population, right? So the vaccine is the right one to come. That's a good stem cell might work, but at the same time, again, the cost is the matter.
Yes, sir. Point is taken. Nalini, madam, I think you have
Yes, sir.
Please go ahead
Yes,
with your
sir.
questions,
Can you hear me? Okay, sir. It's a common question which we have always have a doubt that there are many reports that male is more infected than female. But in today newspaper, I had read that in Chennai, female is more infected. So, is there any study regarding the pathogenesis and sex?
So I think I had a slide to show in China initially. There is no difference between male and female until that March time point, but after that, it is different in different part of the world. Particularly US, we have more of males infected than females. We don't know the host response behind it. So at this point, we don't know the strategy. What's happening. As I said earlier, the studies has to really come and give those answers.
Okay, sir. Thank you. And as many academicians are attending this conference, so I'd like to post a question that what type of researchers we can focus regarding this COVID-19? As a researcher, you can post some ideas regarding the research, sir.
Up to this point, we have to do zero prevalence first. The first thing that we should know is how many people are infected. Okay, or at least at this point, we should do that quickly to make sure that we detect a lot of people to show how much infection is there. Then only we can throw up some strategy to take those population away from the regular general population. Maybe we can quarantine. We can give them whatever the therapy that is available right now, and then make sure that they are actually isolated. But then only we cannot. Then only we can actually stop the spreading. If not, it's going to spread. So we need to be careful. The best strategy right now is this, and at the same time, the private and then the public and private sector has to come forward to fund scientists to make vaccine or therapy. That is very important.
My another question is, according to your view, how long will it take to release the vaccine, sir?
So it will take 8 months.
It will be successful, or what is the opinion?
So it will take 8 months to 12 months, but at the same time, US is trying to do fast track these things. Right now, most of the vaccines are in phase 2. They have to go phase 1, phase 2, and phase 3. So they actually cross the safety phase 1 safety thing. Though probably it will take another 8 months to 1 year, but 12 months. But probably if it works well, and then if they fast track it, probably it may come even sooner. But at this point, we cannot pinpoint it's going to come in this month or so.
What will be the success rate of the vaccines?
We don't know that, right? Like people are in phase 2 trial right now. So phase 1, phase 2, phase 3. The difference between these three phases is the number of individuals getting tested. So we would have to see how the efficacy is between the large population. Once we see a good efficacy, probably that might determine everything. So people are doing it. Once we get a good knowledge about that, probably then only we can answer to that question.
The question is, some asymptomatic people are those. They have more immunity than symptomatic people. Asymptomatic people, do they have more?
That's a good question. There is no right comparison right now, as shown clearly between asymptomatic versus symptomatic. But the problem, we can say that the convalescent people might have a good immune response than the chronic, than the severe patients. At this point, again, we are in the learning curve right now. We don't know the answer right now. So, but that's a very good question. So,
Nalini, madam,
Thank you.
you have more questions, madam?
Sir, sir. I know, sir, enough, sir. Thank you for.
I think we have some more questions left in the YouTube group. In fact, one of the ANJAC alumnus, who's right now with the BD Laboratory, Baltimore, US, wants to ask you, is there a role for...
what's his name? Kalyan Kumar?
You should be knowing Kalyan, right?
Yeah. She was classmate.
Oh, okay, fine. So he is asking, can you please let us know what is the status of the disease targets like IL-17? Is there being considered at all, or is there any research which is happening on IL-17?
Right. So I think Kalyan is my very good friend actually. So Kalyan, right now people are trying to look at different aspects of it. Again, right now I think it looks like they were trying to put down the IL-6. They're trying to block the IL-6, and then people are trying different strategies to block different cytokines. In fact, to block the cytokine storm. Looks like that the IL-6 is the major thing that actually popping out in different studies, that these people have a lot of IL-6. But again, the IL-17 story, very few people have shown that IL-17 elevation in this particular patients, but then we don't know what side of IL-17 is actually making the cytokine storm worse. At this point, there is no much concrete data on IL-17, but IL-6 is the one that people are trying to block right now.
Thank you, sir. There is one very, I would say, thought-provoking question from one Dr. Benazir Fatima. She goes on to ask, sir, is there a life cycle period for COVID-19 virus? So is it a self-limiting, or does it have a life period of survival?
At this point, I think, again, when you look at the infection curve, it stays on for like 14 days or something. Again, the data that is available with different sampling method and different parts of the body, for example, if you look at the monkeys, as I showed now in the monkey trial, whatever they published from Oxford, where they have the virus, they don't have the virus in the bowel, but they have the virus in the nasal cavity. So at this point, even to determine the life cycle, the whole cycle is also it's not been concluded yet. So again, to answer, this is a brand new virus. We just exposed to this virus just 5 months. Still, a lot of data needs to be coming in order to determine all those stuff. But it's a good question.
And there is a question from Dr. Jennifer Jebakruba. Sir, COVID screening has been done with various methods. Is there a role, or can we consider using loop-mediated isothermal amplification? It looks like it is cost-effective than RT-PCR. Do you do that? What is your take on it?
We don't do that, but I don't know much about that particular method at this point. So,
and then one question from Dr. Madan Nals. She goes on to ask you, sir, there are many targets which have been worked upon globally. Is IL-12 being considered for research? Do you have any information whether to tell IL-12 gets activated?
So with the initial response, you have to have IL-12. Without IL-12, a good adaptive immune response won't get generated. Okay. For instance, I am trying to do an IL-12 therapy for HIV. I got a grant on that to do it. But then you need to have a good B cell response. For that good B cell response, you need to have good IL-12 response. if you, I don't think the IL-12 is doing the cytokine storm. No, I don't think so, because that actually elicits a good B cell response. So IL-12 might not be the one that is actually using the cytokine storm. I don't think that is doing it.
Then there is a combined question from
there is evidence, that for a good T cell or a B cell to work very well, you need to have a coordination of a B cell and a T cell in a very good way. For example, in a T cell has, particularly for the CD8 T cell, the CD8 should have perforin and granzyme, the killing potential very well. Then only they can kill. But in this particular cytokine storm, people looks like they are low. They have the low level of perforin. So, meaning you have a CD8 T cells without a functional capacity. So, when you have IL-12, I know for sure when you give IL-12, these particular cytolytic molecules like perforin, granzyme actually elevated in the CD8 T cells. I think IL-12 might not really cause a cytokine storm.
Point is taken, sir. There have been a combined question.
It's a combination factor, sir.
I understand, sir.
You see that, right? Like again, this is a receptor level, also very important. Different tissues has a different receptor expression of this particular virus. Looks like it's very systemic. It's the virus. It's actually in the body. That's not like concentrated only in the lung. Of course, it's concentrated in the lung, but it's also affecting heart. It's also affecting kidney. Kidney express high levels of ACE2 receptor. So the first thing, right after the lung get infected, and the organ failure would be the kidney. So there are a lot of combination factors that's happening. And again, just with the serology, looking at certain cytokines, we cannot determine, this is what happening globally. We never had a tissue-based work. So animal studies are really important to ask these type of questions. Then only we'll know to answer certain questions like IL-12, IL-17, something like that. So that's my take on that.
Sure, sir, sure. And there is a combined question from Dr. Pavitra Suresh and Dr. Saravanan. I think they're treating physicians, and they'd like to know, sir, on what basis the protocols for treatment are being evolved. Is it experience based or lab input based? Of course, clinical outcome also is considered for ongoing continuation of therapy or discontinuation of therapy. Is there any lab inputs which have been very critically used for managing clinical therapies?
No, of course. For example, if you look at remdesivir, they showed this effective treatment in mouse model, in vitro system, as well as in monkey model. Then only actually got attention, and then it was tried in human right now. So again, you would have to test this thing in the preclinical model and animal models, and particularly the animal models are very useful for this. Then only we can actually go for the human trial. I think any drug that actually that we are planning to test, probably have to go to in vitro system in animal models for the safety, and then probably after that, probably the safety is fine, then we would have to go to the human model. So I think remdesivir is, people are trying to recommend that, I guess, in US. Again, they also right now trying to do with the combination therapy to see whether, let's say like a plasma therapy plus the remdesivir, or a B cell therapy, the antibody therapy plus remdesivir, probably that may work. They're trying to look at the different combination, particularly for the critically ill patients.
I think questions are flowing in, but then I think we have time limitations. I have two important questions to you, Dr. Vijayakumar. We've been talking for almost now 6 weeks about various issues. The China model, especially, they were bold enough in restoring the lockdowns, and of course, few sporadic cases are being reported that they claim to tell they are imported cases and they are not locally driven. And the most important challenge governments want to know is there a shortcut for herd immunity so that we can declare people cure and resume normalcy? But China definitely had a very bold approach Unlike a few states in US have opened up, and few states are still very, very tightly locked down, or so much of distancing. In spite of economic challenges, few state governors are very, very, very skeptical about opening up the country back, or opening the economy back, in short. But the China, and that point of view, went on as a bold move. I would like to know, is there a model which they are trying to work on, and they say, okay, fine, Chinese are safe? Or is there any big data analytics? I think we, like some time back also we discussed, the big data is giving a lot of input about the spread pattern, and the especially the data is being to point out, even though for contact tracing it is being used, what contacts are going to be turning positive is also being suggested by that. Do you have any thoughts, or do you have any work which is being happening in your place in regard, so that it helps the policymakers to open up the economy as such?
Uh, it's a complicated question, but what I feel, uh, from the time of infection happened versus the time they actually isolated the virus, actually China did a good job in sending the sequence results right January 10th. Okay, but then they also had a difficulty in controlling the infection because it's actually community spread. And then after Feb 27th, the whole thing came out to the country, like outside China, cases started increasing, and then the deaths are going down in China. But I don't know what exact exit strategy they're trying to do. But then looks like they are still staying in like 80,000 cases. For a while, it has been 80,000 cases. We don't know until or unless a clear study report coming from China. We cannot say that. At the same time, I don't know. I can't answer the second part of your question about the exiting the lockdown system. But what I can caution, particularly in India, is that if you exit those lockdown system, I know the essential workers is important. At the same time, we would have to really take care of people by not spreading this infection. So we'd have to come up with a strategy to make a balance of economy as well as treating this particular infection. So I think we'd have to have a strategy of releasing the lockdown for certain sectors, that is really essentials. Again, when I gave a talk last time, and I got a comment saying that there are people who has money can stay at lockdown. People don't have money, they don't they can't afford this much lockdown because they have to go out there and get money. I'm also, my family is also a part of that type of family. But then when I think about it again, we would have to do something, and then the government has to come with a beautiful policy so that it will help everybody moving forward. At the same time, what I feel is the stigma. We should clear the stigma out, okay? And we shouldn't be looking at certain people, oh, they are COVID positive and all. We should take that away from that. At the same time, as much as possible, we would have to educate the people who don't know about COVID-19. We should go and tell them what is there and what could be the reason that they can, what would be the strategy for them to really get cured and all those stuff, and that will help us. For example, when I heard that the doctor has been beaten up by a general public, it bothers me. I thought of writing I was in fact contacting a couple of my juniors to write letters to editor to Hindu to see whether we would have to really, as a scientist, as a known people who works on this virus, probably would have go and tell people that this is normal. You have to handle this as a regular thing. Don't get panic, and then we have to educate them. Until or unless this won't happen. The people will be very much will be in stigma, and then everything will fall in a different place. We would have to educate them. That's the main thing that we should do. And again, we would have to have a strategy for trying to trying to get rid of the virus as much as possible. We would have to try.
And to close down one truly technical question, I really don't have an answer. You share your thoughts. So a lot of research has been happening from now the mapping of virome is happening in a big way. From our perspective, the microbiome based approach are going to be the future of medical care for various medical ailments.
Yeah.
I'm sure the cold specimens, especially from the Alps and from the Arctic region, and the deep sea drilled specimens are being pulled out for understanding the disease pattern earlier. In fact, I was told that few of the mummies' abdominal contents and through, I think one from UK, if I remember right, bang in the London, the brain of a particular corpse was fully available for sequencing and other things. Do you have any thoughts or do you have any information that such type of viruses, mainly corona or may not be corona, have, but I'm sure we have the Spanish flu experience in 1920, we have clear-cut documented historical controls. Undocumented historical viral pandemics, do we have any, I would say, paleo research which is happening or which has shown some light, so that do we have any learning from that type of virome research?
So that is a very good question, and I also have an interest on it, particularly when you talk about the microbiome. We are also trying to do, we have right now, we're trying to look at microbiome in HIV infected, meaning monkeys, and I'm trying to do something on COVID-19. I'm talking to people from New York, trying to get some data on the microbiome. I think when you talk about microbiome, this is a huge area. Nobody knows clearly what pattern exists. Again, we'd have to study certain things. For example, I came across a study where, as you know, the pancreatic cancer is one of the worst cancer in the world. And then that study tells that in the oral microbiome, the bacteria that is present in the oral, in the mouth, actually seen high in the pancreas. So I was talking to one of orthodontic person here in Atlanta to see a collaborative study to see whether that pattern is actually really happening. And then again, the flossing is important. When you do a flossing, that then you get rid of most of the microbes. We don't know which is good and which is bad. And again, this will happen in different levels. Virus can influence the microbiome, immune response can influence the microbiome, food can influence the microbiome, environmental can influence the microbiome. So since microbiome influence the immune response as well. There are studies, if you deplete certain types of bacteria, you don't get a good immune response. And I was always cautioned, if somebody takes a vaccine, but don't take antibiotics before that. Probably, again, if you take antibiotics and if you get rid of your microbiome, particularly the gut microbiome, it will take 1 year for you to get the same amount of microbiome. So I would caution people not to have, we don't know right now, but at the same time, don't deplete your good bacteria. So again, sir, to answer your question, there are a lot of research going on, and we were also trying to, I'm trying to do that as well in HIV, as well as for the COVID. Let's see what we can get. Now, again, the microbiome in the lung is different from the gut is different from the oral and the skin and all the stuff. We would have to have again, we need a lot of community-based studies and then focus studies on different populations. Microbiome is very different in certain ethnic groups, okay, because we eat completely a different food pattern compared to the Western countries. So again, we eat a lot of spicy and a lot of vegetables compared to other people. We might have a different microbiome, or probably a good microbiome. Again, the curd, one of the good bacteria is lactobacillus. We eat most of the curd as well. So we don't know what is what is actually regulating all these things. It's a combination factor, but again, we need a huge funding to do a massive study. And one of my friend, his name is Bharat, He's in New York, and I'm trying to collaborate with him because he's from the top microbiome lab. He's trying to help me for this. Again, a lot of various factors.
Too early to correlate or extrapolate towards COVID, I guess. So I think we are now coming to close. Professor Nalini, ma'am, do you have any questions left with at this point of time?
But microbiome does influence a lot of things. So microbes is the world, they make us. It's not like we are making us. Actually, that's important.
Point is taken, sir. Nalini, madam, do you have any more questions, madam?
Yes, sir. I do have a final question. Since Vijayakumar sir is working in HIV vaccine, I want to ask sir related to HIV. But sir, is HIV vaccine commercially available for all patients? What about say something about this HIV vaccine status, sir?
There is no commercially available HIV vaccine. Everything is in the preclinical trial in the animal model. There is no any vaccine for HIV right now. So all are trying different ways. We are trying DNA MVA based vaccines, and again we are also trying protein based vaccines, and different people are also trying ad based vaccine, CMV based vaccine, T cell vaccines, and then now people are trying to use a protein, sourcing protein as a vaccine. So we all are trying to make a good neutralizing antibody in a combination with a good T cell response. If you a couple of days before we published a group of, I'm not again a part of it, but I'm doing a different aspect of vaccine here. We published a data showing that protein vaccine is really good for eliciting a good neutralizing antibody. That's in Nature Medicine. If you go to PubMed and look at Rama Amara, probably you can see that paper. So that is again in the monkey model. But it's one of the best paper, and people are different. When Scripps is working on a good vaccine, and Harvard is working on a good vaccine, like Dan Baruch, they're working on vaccine, but they all are trying in the animal models to get a good neutralizing antibody. So again, vaccine for COVID is not vaccine for similar like vaccine for HIV. This virus is mutates like anything. So it's for 30 years we don't have a vaccine for this. We've been funded a lot to get a vaccine, but we are still struggling to get a vaccine, meaning this virus mutates drastically, not like COVID, so that's the reason we take some time. At the same time, this particular virus once they deplete the central architecture of T cell, that is a CD4 T cells. So when you lose your CD4 T cell, then you lose a complete immune response. So this is a matter of that window period of the infection versus take on of the virus. By that time the vaccine has to work. That's what we are trying to make. Within that time, we would have to fully have a good immune response, like particularly good B cell response, assured with a good T cell response. So that's not an easy task.
Thank you, sir. Thank you, sir, for your nice answer. Another one, Sendil, sir, do you want to ask any questions? Sir, can you unmute Sendil, sir?
Sendil, I was doing my MSc, he helped me a lot.
Sendil, sir, please unmute yourself, sir. Sendil, sir.
என்னுடைய கொஸ்டின் எல்லாம் கேட்டாங்க சார், ரொம்ப தேங்க்ஸ்.
I think today's discussion was extensive, but still it is our responsibility. I think I missed your name. Very sorry, sir.
No, sir. No, not necessary, sir. No problem.
Madam, anything else, madam?
Nothing, sir. We can conclude the session, sir.
HOD, sir, your closing comments before we move on to Shivakumar, sir, for vote of thanks. HOD, sir, please. Yes, sir.
Hello.
You are with us, sir.
Hello.
Yes, sir. We are able to hear you, sir. Pesanga, sir.
Okay, sir. Okay, sir. Sir, we can conclude this session, sir.
Thank you, sir. Thank you.
Okay, sir.
Shivakumar, sir. Vote of thanks.
Shivakumar, sir.
Vijayakumar sir was instrumental in getting connected to Vijayakumar Velu. I think they
Yes,
were
sir. Yes, sir.
Yes, sir. Yes, sir.
over to you, sir.
Yes, sir. Okay, sir. Sir, good afternoon to all. First of all, I thank our management to granted to conduct this meeting with a successful one. Then I thank our principal who motivate to conduct this program. Then I thank our today's chief guest, Dr. Vijayakumar. He gave wonderful ideas about COVID-19. Yes, the past, present, and future. So, what is the status and other things, mode of infection and source of infection and treatment prophylaxis? Everything is good. He explained everything is good regarding COVID-19. Thank you, sir. Then I thank our respected Kanagavel sir, Mr. M. Kanagavel sir, who is a member in the Medical Microbiological Association and laparoscopic surgeon, gastroenterologist. He is a wonderful human being. Who have spent a lot of time for conducting this program with a successful one with live YouTube and Zoom. Then I thank our, thank you, sir. Then I thank our head of the department at Ponmanikam and organizing secretary at Nalli madam and other faculty members who make arrange a successful one. Then I thank the participants of various scientists, various institutions of scientists, research scholar, teachers, and student friends. Finally, I thank one and all. Thank you, sir.
Thank you, Shivakumar sir, for connecting Vijayakumar sir with all of us. And in fact, I should thank the two important senior faculty from the Pasteur Institute and the BCG Institute for staying with us all through. Thank you, one and all. And Vijayakumar sir, with your permission, I call this session as end. HOD sir, Nalini madam, can we call the session to end, please?
Thank you all, and thanks for Shiva.
You can end the session, sir.
Thank you all. We will meet next meeting. Thank you. Thank you.
Thank you all for your active participation. Thank you all.
Right.
Shivakumar sir, thank you, sir.
Thank you, madam. Thank you, madam. Thank you. Yes, sir. Thank you. And other faculty.
We can leave the meeting, sir.
Kanagavel sir,
Thank you, sir. Thank you,
sir. Thank you. Kanagavel sir. Vijayakumar sir, Papa, we will meet tomorrow.
Thank you, sir.
Okay, sir. Thank you.
Vijayakumar sir, you wanted to ask something?
No.
Vijayakumar.